Microbiology and Parasitology. 1. Microbiology. Definition of microbiology. Microbiology is a branch of biology that studies microorganisms, including their structure, function, metabolism, pathogenicity, and interactions with humans, animals, and the environment. Branches of microbiology. 1. Bacteriology. Bacterial structure, cell wall, gram-positive, thick, dominant peptidoglycan. Gram-negative, thin, with an outer lipopolysaccharide, LPS, layer. Flagella, used for bacterial movement, motility. Capsule, protects bacteria from phagocytosis. Endospores, defensive structures allowing bacteria to survive extreme conditions, e.g., Clostridium. Bacterial metabolism, obligate aerobes, require oxygen, e.g., Mycobacterium tuberculosis. Obligate anaerobes, cannot survive in oxygenated environments, e.g., Clostridium tetani. Facultative anaerobes, can live with or without oxygen, e.g., Escherichia coli. Bacterial toxins, exotoxins, secreted toxins with specific effects, e.g., diphtheria toxin. Endotoxins, LPS components of gram-negative bacteria released upon bacterial death, e.g., Escherichia coli. 2. Virology. Viral structure, genetic material, DNA or RNA. Capsid, protein coat surrounding the genetic material. Envelope, present in some viruses, derived from the host cell membrane. Virus classification, based on genome, DNA, e.g., herpes simplex, or RNA, e.g., HIV. Based on replication, lytic viruses destroy host cells, or lysogenic viruses integrate into the host genome. Virus replication cycle, adsorption right pointing arrow, penetration right pointing arrow, replication right pointing arrow, assembly right pointing arrow, release. Examples: influenza virus uses hemagglutinin, HA, to bind to host cell receptors. HIV uses GP120 protein to bind to CD4 receptors. 3. Mycology. Fungi, fungal structure, unicellular, candida, or multicellular, hyphae, mycelium. Cell walls made of chitin. Classification of fungal infections. Superficial, infections of skin, hair, nails, e.g., dermatophytosis. Subcutaneous, infections beneath the skin, e.g., sporotrichosis. Systemic, infections of internal organs, e.g., histoplasmosis. 4. Basic immunology. Nonspecific, innate immune response, phagocytosis by macrophages, neutrophils. Complement proteins that destroy microbial membranes. Specific, adaptive immune response, Humoral immunity, antibody production by B cells. Cellular immunity, T cell activation to destroy infected cells. 2. Parasitology. Definition of parasitology. Parasitology is the study of parasitic organisms, including their classification, morphology, life cycles, infection mechanisms, as well as the diagnosis and control of parasitic diseases. Subdivisions of parasitology. 1. Protozoa. Characteristics of protozoa. Unicellular eukaryotic organisms. Move using flagella, cilia, or pseudopodia. Examples of key protozoa. Plasmodium spp. Causes malaria, transmitted by Anopheles mosquitoes. Entamoeba histolytica, causes amoebic dysentery. Pathogenicity of protozoa. Invade tissues or blood cells, e.g., Plasmodium destroys red blood cells. Release enzymes that damage host tissues. 2. Helminths, worms, classification of helminths. Nematodes, roundworms, Ascaris lumbricoides. Cestodes, tapeworms, Taenia saginata. Trematodes, flukes, Fasciola hepatica. Life cycles, direct, does not require an intermediate host, e.g. Ascaris lumbricoides. Indirect, involves intermediate hosts, e.g., schistosoma. Pathological effects of helminths. Intestinal obstruction by Ascaris. Anemia caused by blood loss from Ancylostoma duodenale. 3. Arthropods as vectors. Examples of key vectors. Mosquito Aedes aegypti. Transmits dengue fever. Mosquito Anopheles. Transmits malaria. Sandflies. Transmit leishmaniasis. 3. Diagnosis of microbiology and parasitology diseases. 1. Microbiological methods. Gram or Ziehl-Neelsen staining for bacteria. Culture on specific media, e.g. McConkey for gram-negative bacteria. Molecular tests, PCR, for detecting viruses like HIV. 2. Parasitological methods. Stool examination for helminth eggs, Ascaris, Taenia. Peripheral blood smear to detect Plasmodium. Serological tests like ELISA to detect toxoplasma antibodies. 4. Control and prevention. 1. Sterilization and disinfection. Autoclaving medical instruments. Disinfectants for environmental surfaces. 2. Vaccination. Vaccines for dengue fever, hepatitis B3. Vector control. Eliminating mosquito breeding sites. Using insecticides or bed nets. 4. Personal hygiene. Hand washing, proper food preparation to prevent parasitic infections.